A very good evening to our dog and to all my fellow friends. Today we will present about dermatophyte. Our species that we choose is Tricophyton verrucosum. We are from group 5 and group 5 is consists of me, Leobrita Masalon and Darren Chu. So first and foremost, the introduction. Dermatophytosis is a fungal infection caused by dermatophyte that invades the outer layer of the skin, which is stratum corneum, and can pose a major health risk in both animals and human due to its zoonotic potential. Tricophyton verrucosum is common cause for dermatophytosis in cattle and is linked to human infection, especially among farm workers, according to Papini et al. 2010. Calves are more susceptible than adult cattle due to lack of acquired immunity. The transmission occurs via direct contact with the infected animals and with indirect contact, example like contaminated environments, poor management, and also hygiene. According to Agnetti et al. 2014, the disease is more common in hot and humid climates. So, Tricophyton verrucosum also affects other ruminants like goats, sheep, and camels, according to Pierre et al. 1994. And human infection typically involve the face and also the body, but can be treated with antifungal agents, while treatment in cattle is more challenging. Min et al. 2006. So for the objective in this paper, first is to study the zoonotic potential of Tricophyton verrucosum. Second is to understand its role in causing dermatophytosis in animals, especially cattle. Third, to study the factors that influence the infection and spread and assess the risk of transmission from animals to humans. And lastly, to understand its public health significance. So we will be start the discussion. For the first one, types of sample to be taken. We will take skin scrapings and hair from the active border of the lesion because it contains the most fungal elements. And then the sampling technique for this. Before sample collection, the lesion area should be properly cleaned using 70% ethanol to eliminate any surface contaminant. Then the sample can be collected by gently scraping the skin with sterile blunt scalpel or plucking hairs using sterile forceps, according to Guo et al. 2020. Then the collected sample should be placed in sterile dry containers like sealed plastic bag to maintain the sample integrity. So in order to avoid contaminant and overgrowth by other microorganisms, sample must be stored and transported at 4 degrees Celsius. And then we move to direct microscopic examination. So a portion of the sample is placed on a clean glass slide with a drop of 10% potassium hydroxide or KOH, which helps to dissolve keratin and debris for clearer visualization of fungal elements, according to Dalis et al. 2019. Then a cover slip is gently applied over the sample for a few minutes before the microscopic examination. Then, lactophenol cotton blue staining is used to stain the hyphae, spores, and also the canidia. According to the findings of Dalis et al. 2014, microscopic examination of isolate stain with lactophenol cotton blue revealed a septate hyphae with numerous clavate microconidia born laterally from the hyphae, some of which were breaking into arthrospores. So this is the picture of the macroconidia. So the first figure, we can find the small clavate microconidia laterally attached to the hyphae. You can see from the arrow. And then the next figure, we can find a single longish, thick, small wall smooth wall, and multi-septate macroconidia with characteristic ray tail, which is the string bean shape, typical of Tricophyton verrucosum. This is the picture of it, and it is from Dalis et al. 2014. Both of the picture is from Dalis et al. 2014. Then we move.
Okay, so now it's for the fungal culture of T. verrucosum. So to begin the diagnostic process, we cultivate the suspected fungi sample using Saborodextrose agar or SDA. This medium is ideal because it has high glucose content and is slightly acidic, which is favorable for dermatophyte growth. Alternatively, we can also use DTM or dermatophyte test medium, which includes a phenol rate, which is a pH indicator that changes color from yellow to red in the presence of dermatophytes. This is due to the byproducts of dermatophytes being more alkaline. It also contains antibiotics to prevent bacterial contamination. And depending on the suspected species, specialized Tricophyton agars can also be used for more specific nutritional profiling. It's important we collect the samples with such as skin scrapings and hairs and nails sterilely to prevent contamination. And incubation takes place at about 30 to 37 degrees Celsius because it is more thermophilic and it grows slower, which is like 1 to 2 weeks for the full growth. Next. Okay, now for the microscopic examination of the culture, we we use the native staining. So after sufficient colony growth, we perform microscopic examination. This is done through a wet mount or teased mount using part of the fungal colony. Dyes are placed to enhance contrast. For example, we can use chalcophore white, which binds to the fungal cell walls, and fluorescence under UV light, making it great for observing structures. Another one is lactophenol cotton blue (LPCB) is another common choice as it both stains and preserves fungal features. Using the 40 times objective lens, we look for characteristic traits for T. verrucosum, which will be discussed later. And for the reader method, the reader slide culture is especially useful when we want to observe fungi with in their natural arrangement. So this setup involves using a block of agar, like cornmeal agar or PTA, on a sterile slide over a U-shaped rod and moistened filter paper. When inoculated, the agar block will with fungal material and gently paste over a cover slip on top. This is incubated for 1 to 2 weeks at 30 to 37 degrees Celsius in a humid environment. And during growth, fungal structures were spread between the cover slip and the agar. After incubation, we carefully lift up the cover slip and stain it for microscopic observation. Okay, so these are example of microscopic examination taken from journals. So as you can see on the left side, it's a string bean shaped macroconidia and terminal vesicles at the tips of hyphae and chains of chlamydoconidia is on the right side. It looks like chains. And on the black and white picture, this is from scanning electron microscope. As you can see, for Tricophyton verrucosum, there is intermediate chlamydiospores of arranged in bead-like formation, and the bottom one is just a comparison. The bottom one is spherical or ovoid microconidia from Tricophyton mentogrophytes. Okay, another one. This is for the macroscopic examination using, as you can see, the picture A and picture B, which is the white one. A, it looks like a button or dish shape. Its color normally white to cream colored fungal colonies with a raised center and a flat periphery. For B, picture B is a microscopic one. It is broad, irregular hyphae, and the chains of chlamydospores referred to as chains of pearls because they are in a chain as a key microscopic feature for the fungus. And for the reader method, this is just an example. So there are many methods to do it. Some use a U-shaped glass rod, like the top picture, and some is just a block of agar with the microscope slide placed on top of it. Okay, so for the results, identification results, the final identification involves comparing both morphological and microscopic features. So, Tricophyton verrucosum is recognized by its sparse, irregular macroconidia and chlamydospores chains. This in contrast to metagrophytes, which produces a spiral hyphae, and if you can compare it, the colony morphology is also a key factor. So T. verrucosum produces a small waxy colony that appears white to cream on top of a and yellow underneath. So, however, to definitely identify the species, molecular techniques such as PCR or ITS sequencing can be used. Okay, conclusion. So, in conclusion, to accurately identify Tricophyton verrucosum, it requires multiple step processes. Culturing the sample gives us
Darren, sorry, suara kau ndak dengar.
Hello, hello.
Halo? Okay.
Oh, nak start lagi dari conclusion atau apa?
Start dari conclusion lah.
Okay. In conclusion, accurately identifying Tricophyton verrucosum requires a multi-step process. Culturing the sample gives us initial growth characteristics, while microscopic exams, both direct and via slide culture, reveal defining features of the fungi. For the most accurate diagnostic, combining morphological data with molecular tools such as PCR will ensure distinguishing Tricophyton verrucosum from other Tricophyton species. This approach is essential, especially in the veterinary settings where proper diagnostic leads to effective treatment and infection control. These are the references for the picture. And thank you.
Okay.
Do you have cat cup?